So hello everyone. As you know, I'm Subhash. I recently moved from US to Ahmedabad University. So right now in a phase to establish my lab. So the progress is we can breed the flies now. We started getting my natural populations and now we'll be doing some work. We'll be following a few older work and we'll also touch base some newer ideas and all that. So basically I see physiology in light of evolution and I work at the crossroads of ecology, evolution, and physiology, and more inclined towards the physiological questions. And I use flies as a model system. So, water is an important parameter in our life, and physiologically it is also important. So when we came out from the oceans and tried to settle down on the terrestrial life, so the challenge was how to conserve that, because we know that water is a very crucial element of our body, and when it goes down a certain percent, our physiological system starts collapsing, and at the end, animals die. And on the planet Earth, if you see that, like today, like 30% of terrestrial, environment is arid, and that percentage is going to increase, like due to anthropogenic reasons and all that. Then we came to the, to the terrestrial life that other, some physiological, constraints were there, because so smaller organisms like insects, they have a smaller size, so they have another challenge because they have lots of open surface area and through which water can easily get away. So if you see here, the flea all the way up to the humans, and if you see that, that water loss rate, it's way higher here in the smaller organisms when you compare with those bigger organisms. And the simple physics is right here on the left panel. So the water has a very, unique kind of tendency. It's more active inside, so it always has a tendency to escape from the body. So it's a chemical nature. So if you take like this beetle, a small amount of water can easily get away. And when it reaches to a certain level or certain percentage, the animal's physiological system collapses and the animal dies. And like in insects, people have been working for like last 50 to 80 years, and they have figured out that water can lose through various ways, and insects can develop some sort of kinds of strategies. And so for whatever they have figured out, there could be three possible ways: by increasing the amount of water in the body. So there's some organisms have like tendency to have more water in their body than the other organisms. The second thing, tolerating the loss of greater percent of body water, like dehydration tolerance, that might vary, like species to species or within populations. And then the third one is reducing rates of water loss, where most of my work is, centered, and I'm very much interested in, how organisms, manipulate these rates and how they conserve water. There is a very important reason behind that. So if you take this as a simple fly, and if you see, hardly 20%, organisms lose while egg laying or through the oral secretions or the excretory material, etc. So the remaining 80% they lose through this hard cuticle. It's more or less typical for most of the insects. So it's like in a significant amount of water they regulate through the body cuticle and those spiracles. And even if you add those spiracles, although they are located here on these each tergite ends, they hardly, like in flies, constitute like 6 to 7%. So everything, including this 20% plus respiratory and all thing, like comes around 93%, like 17% or something. So like particularly like melanogaster, it regulates like 93%, like almost 83% of water through this hard cuticle. So it's an important, physiological characteristics. And I'll try to touch some of these organism levels, like genes, and at the population levels, and how, at the environmental level, how environmental parameters are affecting these populations and how they end up, getting some sort of different genetic background and whatever. So we'll be mostly, this talk will have three separate components. One where I will be introducing you with some natural variations, like along latitudes and altitudes. So my work starts from the field. I go to the field, collect natural populations at different latitudes and different altitudes, bring them to the laboratory and do common garden experiments. So where I expose these populations coming from different locally adapted conditions and then put them at a common condition like 25 degrees centigrade. And here, okay, so, hello? Can you hear me now? Okay, so, for example, you collected like 8 or 10 populations all along Indian latitudes, you bring them to the laboratory, subject them to 25 degrees centigrade, and assay them. So it's like these are very, typical structure kind of experiments people have been doing, those evolution people in the laboratory to, find out those, environmental stresses, potentials in the population. Then second section of my talk will be like whatever patterns you see in the natural populations, you try to, sometime, test those hypotheses, the kind of patterns you have seen along latitudes and altitudes, and how will you test these hypotheses in the laboratory? So I use artificial selection to address such questions. So I will also touch base some small section where we tested a hypothesis, and then finally we will get into the genomics, where we did a GWAS using those DGRP panel. Those who are working with Drosophilids are already familiar with those. Who are not, I will give you some brief intro for that. Okay, so as we all know, Drosophila melanogaster, the model organism I'm covering in today's talk, originated somewhere here in Africa. And from there, they colonized like pretty much to every part of the globe. And they pretty much followed, this all happened in the last 10,000 years. And like in Australia and in North America, they entered just like in the last 300 years. So like pretty much it shows like they went with the humans. Just like human commensal, wherever humans went, these flies went with them, with their fruits or whatever, feeding stuff and all that. So again, it makes an fantastic example to study like evolution at a faster rate, like in Australia and Northern America, than like in India, like 10,000 years in these other places. So like you can directly compare, faster pace versus like a slower pace or whatever. Yeah. So other way around, quite possible. But I'm just giving you an example, from where they started and where they settled down. So all these was through the ships. So if ships were going that way, so someone has to test that. Okay. So they might have to, find out, it needs lots of structuring of populations at the local conditions here as well as here, and then, these parts. But right now, we are just focusing about this particular, pattern. But this is just to give you a background information. This is not directly related to the kind of thing I'm showing you today here. Okay, so when it comes to the natural variation, so most recent work, we collected these 6 populations all along the east coast of the United States. And we exposed these 6 populations to 3 different temperature conditions, and particularly females were exposed to this low temperature and short photo period. So in temperate world, what happens? This is very easy. So we wanted to test the thermal response for these populations collected along the latitudinal cline, and also because in the temperate world, the tropical flies are different from the temperate flies in few cases. One is like diapause. So like flies which are living way up here in the colder climate, usually before the onset of winter, their ovaries shrink, and during that entire snow period, they don't lay egg. So it's like rechannelizing the resources and all that. And then, so that population, goes near to extinction, like hardly 5%, remains, and they reestablish during the next spring and expand their population size and reestablish. So this like new phenomenon is coming, that this seasonal dynamics, pretty much is establishing these cline of variations and all that. So we brought back, and what you see here, these dotted lines, females, solid lines, males, and this extra line is for female diapause. So if you see, like, as temperature goes down, they are increasing their resistance for the drought tolerance. So all these populations were exposed to drier conditions. So creating an artificial desert in a while is very simple. You take some silica gel, put a styrofoam, and transfer your flies, and you keep it separated, aerated. That silica gel will absorb entire moisture from the wild, and you have sufficient oxygen like for 8 to 10 flies for next 20 to 30 hours. So it's very simple. So we scanned like 30 to 40 isomorphism lines from each of these, white star locations. And you see in a positive cline, a clear positive cline, that all of those higher latitude flies from the north are surviving longer than the flies which are living pretty much in subtropical kind of condition like Florida, where temperature is relatively higher throughout the year. So these flies are living relatively shorter than these flies. So you see such patterns. Coming back to India. So like India is a very unique, subcontinent where, 17 traits have been characterized so far, and most of them are showing positive or negative cline. So it's a fantastic system in tropical, weather where you can't expect such, surprises, but they are there. So this is all along Indian latitudes, and if you see, there are almost 6 different species, and all are showing a positive cline. All North Indian, Drosophilids are more desiccation tolerant than those southern Indian flies, and this variation is huge. Like along east coast of United States, like flies have 2 to 3 hours difference, but here it's like 6 to 7 hours difference between melanogaster particularly. So maybe because seasonal variations are more intense here, or that temperature on the upper end is way higher than those temperate world. Yeah. Are relative to each other here right now. So we took that actual data. So this is along the latitude for the 6 species. So these different dots are representing those different species here. And this line just, just to show that common trend or pattern. Yes, exactly. Yeah. It doesn't matter. So, so when you see that, want to put that entire, this, geographical pattern under that, climatic variables, we say that, as you move towards north, that seasonal variation gets intensified. And when you compare these, I have just put these 3 locations climatic data. So it's like Trivandrum is right here. You don't see too much temperature variation throughout the year, but as you move towards, northern part of the country, you see that, like Jammu, for example, you see a huge variation like in these 2. Even in the center, you see some sort of variation. So some studies, those climatic model testing and all that have come up with that coefficient of temperature variation. This is like, yearly how temperature is varying, low and high temperature throughout the year. Whatever that variation has, that justifies most of that, pattern. But this is like climatic, correlation kind of studies, and it doesn't tell much. So with these, clients discovered different parts of the globe. So there were some existing data on the cuticular hydrocarbons. So when you see insects outside in the nature, you see their glossy layer. So that's the lipid layer. And that basically protects, because those lipids are hydrophobic in nature. So they have an inbuilt mechanism by which they try to keep that water inside intact. So then the other thing came up, like maybe there are some other characteristics in the membrane which makes membrane impermeable, so that it becomes a hydrophobic layer. And under different given environmental conditions and parameters, that membrane might be dynamic in nature. Like when you compare north, center, and south, there may be some relative changes in that entire cuticular membrane where I'm interested. So here we are considering these some simple, assumption that membrane A versus membrane B. So this is an cuticle, insect cuticle. So if you see it in detail, there is an epithelial layer. This is inside body, and above that layer, there's chitin layer, and then that is overlaid by the cross-linked lipids, proteins, and melanin. So most of your melanin component is here. That whatever color you see here, that's under that cuticular hydrocarbon layer, that glossy layer which you see. So that other than cuticular hydrocarbon, there could be some other potential members, and that could be melanin, because these are 5-ringed amide kind of compounds, organic compounds, which are very rigid and hard to break. And they might be like, when you compare flies, because people have discovered the cline for the pigmentation as well. And if pigmentation and desiccation is going hand in hand, flies which are more resistant to drought tolerance are darker normally. When you compare the flies which are less resistant to desiccation, they are lighter in pigmentation. So we wanted to test that. So here, before we get into that testing, So if you grow flies at a lower temperature, they will end up in getting darker pigmentation. And flies which are growing at higher temperature, they will end up in lighter phenotypes. So they have a very clear temperature relationship, so you can clearly go back to the nature and can try to justify that also. And hydrocarbons have all the way reverse, because when flies are growing at higher temperature, they end up getting longer chain length compounds, and these compounds are very hard to break. So they have an advantage. So like you put fly at a higher temperature, and in real time, if you start accessing the characteristics of the cuticular hydrocarbons, the compounds which sustain longer, those were the longer chain cuticular hydrocarbons. The smaller ones will start breaking very quickly. So like when we characterize, we also characterize those those 6 populations collected along the east coast of the United States. You see a clear dynamic shift in the cuticular hydrocarbon chain length. So more of the flies which were coming from the Florida, they have longer chain hydrocarbons. So, and that systematically changes, gradually. Yeah. 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 Exactly. So you see thermal plasticity also. Yeah. And when you see here, this is, I tried to review that. So this all comes from that Indian subcontinent. So this PZ pigmentation, so you see a cline, yes, but in cuticular hydrocarbons, you don't see a cline. So this is, I'm talking about the total cuticular hydrocarbons isolated from the flies. But sometimes when you go one step further, it's in a relative. So it's in a cocktail of different cuticular hydrocarbons. But here we're just talking about the total cuticular hydrocarbons, because people have studied, those grasshoppers and all that, and they have found that, even the total cuticular hydrocarbon, also changes, among the populations. So here, there were not one case, there were many cases where people also reported pigmentation cline. So then that, hypotheses start coming into shape that maybe pigmentation desiccation have some sort of relationship, and because of this co-varying nature of that pigmentation with the desiccation cline might doing something, making this membrane, behave in a different way. So we wanted to test that. So this was, a gap here. Here we clearly know that cuticular hydrocarbons are, hydrophobic in nature, and people have developed certain models there. They have tested, water, movement and all that. So we wanted to test that whether pigmentation works or not. So typically when you see these, people have been characterizing clines all along the east coast of Australia, east coast of the United States, all along Indian latitudes, and they have come up with these general conclusions that dry and variable climate in northern and southern higher latitudes, northern versus southern high latitudes, so in Australia and other way directions, populations of Sophophora are more desiccation resistant relatively because the clines have been investigated on these parts of the globe, and northern and southern populations are darker. So these are general patterns people have found. So this gave the foundation to that hypothesis. So previously, pigmentation, it does many things because these genes are notoriously famous for pleiotropic activities. So they're doing lots of roles. They are involved in temperature regulation. They are involved in pathogen resistance. They are involved in crypsis, reproduction, and cuticular waterproofing. So this we particularly wanted to test. So what I'm showing you, that's particularly about the cuticular waterproofing. So what we did, we went to the wild. We collected around 400 females. And what we did, we picked 80 darkest female from those 400. And then we also picked 96 light females. So when you scan that population, you see that bell-shaped curve. So we picked the ends for dark and light. And then we kept that one unselected line. So right now it's like around 160 or 170 generation. It started like in 2008 and somewhere. So every time what we do, here, whatever eggs came, progeny came, we again selected like 10% of the darkest. And we allowed to lay eggs to them, and that founded that next generation. Here we picked the 10% most lightest from the population. And these were totally unselected throughout the selection. And density was controlled. So like around 200 eggs, something like that, were picked. So this was in a threefold controlled selection regime. And on the other hand, we had these desiccation selection lines. So where this is started when I joined my postdoc lab. So my postdoc PI started pretty much with the same numbers, 400 females collected from that same geographical region, and then they had 3 desiccation selected lines and 3 controlled lines. And what they were doing, they were creating an artificial desert here in this cage. You take a bowl of silica gel and wait until 85% dies. So get those remaining 51, feed them, and collect eggs from those remaining females. So every time you do this. So we had 2 good comparative, like on one hand, you selected populations for drought tolerance, and on the other hand, you selected populations for the pigmentation, right? So you can directly compare these and can ask some good questions. So like dark selected flies will be more desiccation resistant, and light selected flies will be less so. And desiccation selected populations will be darker than non-selected control populations. So this, if everything goes okay, and if that hypothesis exists, we might see some sort of patterns. But what happened? So here, this is dark line. You see these highly pigmented flies here, and this is the light line, and these are unselected control lines. So we see a clear, change in those pigmentation. And it starts coming, this trait is so interesting that even after 8 or 10 generations in the lab, you can easily see the change in the coloration in the flies if you start selecting them. So here, this is, so it's a grayscale, so read it other way around, okay? So this is the dark line, light line, and control line. So what do you see here? Pigmentation selection does not affect desiccation resistance. So we subjected these pigmentation selected lines for the drought toleration, and we didn't see any difference between control, light, or dark lines. Very upset. Then we, also, let's do one litmus test. So, we subjected these flies to respirometry. So respirometry checks the real-time, water loss rate if it's anything related to the membrane. So what do you do here? You take a fly here in this tube, and the air comes and air goes out. So you retrieve all the moisture from it out, and then you try to catch the signal, how much water is coming out. So you pass the dry air here, and a wet air comes out here. So the sensor gives you that data, and all these peaks are excretion, so you can exclude these peaks, and this, whatever that water is releasing here, you can, integrate this and can get that actual, nanoliter per hours. So measurements, nothing. So pigmentation was doing nothing, particularly in selection lines. So here, desiccation selected lines. So in desiccation selection lines, when you compare those controls, these Fs are controls and these are, treated ones. So they do survive longer. It's like double. So they are increasing their survival hours, they just get double, 15 to 25, 30 hours, something like that. So you see that clear signature for that. But when you score them for pigmentation, you see nothing. So they are more or less equal. So there is no change in coloration in control versus desiccated ones. So through this talk, I just want to give you a flavor that how complicated, the patterns are, what we see in the nature. Sometimes we get success while selecting them, and selection experiments are like this, sometimes we get a better understanding, and sometimes we didn't get the answers, so that, takes us back to that, nature, and that time we realized that, in the natural environment is way more complicated the way we expect them, but these are the ways to test, the things and reject things. So here, a general comparison, image-wise. So desiccation selected and fed controls, nothing. Here you see color shift, and they don't show any difference for that pigmentation, but they do show difference for the water loss rate and even for the drought tolerance. But here they show difference for the pigmentation, but didn't show any difference for the drought tolerance. So dark selected flies will be more desiccation resistant, light selected will be less, so no. And desiccation selected populations will be darker than non-selected control populations, no. But we found something way more interesting, which I'm not going to discuss today. So pigmentation selection lines exhibited unexpected correlated evolution in body size and glycogen content. So glycogen is, it's a organic molecule which can hold 5 times more water molecules than any other, metabolite. So that might be helping these flies. So when we selected this particular, maybe this loci was sitting right next to color or something, I don't know exactly right now. So we're trying to figure it out, what happened, and we are testing this in outdoor cages experiments, and we'll go back to the nature and will do like, yeah. And we are also scanning those DGRP panel for these correlations. Yes. Is by increasing carbohydrates, because in selection lines, people have found that the people selected those, that fed control and desiccated lines. So desiccated populations also have this same pattern. They have more glycogen content. No, it's not the only way. I'm just, touching that third part where I was saying, flies can get more water content than the other was, dehydration tolerance. This is one of the mechanisms where my interest lies because, yeah. So this is one of the mechanisms. So we were just testing, one aspect. Okay, so then, we went to that DGRP panel. So those who are working with flies might be familiar with this. So Trudy Maker, North Carolina, she went outside to the fields, collected like around 200 females, and she inbred them for 20 generations. So after 20 generations, these flies reach to like a near homozygosity. So like these colors, you have different genetic backgrounds, and here, they become independent to each other. And then she sequenced them. And then she created this, this platform. So here what do you do? This platform has in background that 200 those isofemia lines sequencing data. And then what do you do here? You take this panel, entire panel, and you subject this panel for the drought tolerance and assay them, collect the data for males and females, and go back to that, database and subject, or upload your data, and you will get those, possible, loci or SNPs and whatever. So we did that, and what we found, we ended up around these 36 SNPs, and these were correlated with the drought tolerance phenotypic data, and from these, SNPs, we ended up, nearing down to 20 genes and one non-coding regions, and these 20 genes were these 5-HT2, BBG, beta-La, and CZ, unknown, C-trip, EST, and then we took them for some initial level of Go biological analysis, and these 20 genes, easily fit in metabolism, signaling, cuticle development, and transport. So, of course, cuticle is there. So we don't understand that, the dynamic nature, how that cuticle is changing in nature under different environmental and, temperature parameters, and metabolism, of course, because lots of, these flies, when you scan them for protein, carbohydrates, and lipids, these selection lines, particularly for the drought tolerance, you see significant, variations in these three metabolites. And signaling, because that one of the species I have been working, when I was doing my first postdoc, Drosophila mohavensis, it survives on those cactus species in Sonoran Desert, and where we found that when you expose that fly, particularly to the drought tolerance, they stop, some behavioral changes undergo, and flies start, resting. It doesn't, when you expose flies to the drought tolerance, they become agitated, but that fly, intelligently handles it. So it just sits silence for like 20 hours or 23 hours, and then started, responding. So it's like one way, they are just trying to, channelize or rerouting their metabolic rate by behavioral adjustments and all that. And that could be the reason that we are seeing lots of signaling, those genes which are involved in signaling pathways and all that. And from there, few, interesting genes, for example, that 5-HT from that list and 5-hydroxytyrosine. It's in a serotonin, and serotonin is an important diuretic hormone in insect, and that indicates the DGRP lines may differ in urine production. That's we are predicting, which needs to be tested. And then two additional candidates, the CZ4835s, are functionally linked to chitin metabolism, particularly chitin, that the component of the cuticle. And serotonin also mediates cuticular plasticization. And together, these findings suggest that cuticle permeability may be an important factor affecting desiccation tolerance. And metabolic differences also appear to be important, as SNPs mapped to close to these three genes implicated in lipid metabolisms like KLR, CZ1830, and this is 31089. And so we got like few candidates, to play with. And what we were doing right now, in 2014, where the lab where I was working, they published a paper in PLOS Genetics, and what they did, they collected those same 6 populations which I showed you for their drought tolerance. They did a pooled genome resequencing, and they actually wanted to test that how seasonality affect the cline formation and all that. And they ended up, there were around 1,200 alleles which were cyclically changing every year. So their frequency was going down in fall and comes up in the spring, then goes down and comes up. So, and then that also the relative frequency of these, some of the alleles were also changing. So what we are trying to do, we will take some of these candidate genes right now and trying to look where they do fit here in this cline. So that could give that, final, some more, tight connections of the genes with, how they are changing in the natural population. So that we are still figuring out that the way to do that. And the question which we are asking are genes identified among those North American natural populations being associated with desiccation tolerance in DGRP, show increased signature of spatially varying selection related to the rest of the genome. And if yes, then we can do some finer scale or wiser experiments to test those independent genes. I will stop here. Thank you very much for listening. My collaborator is Alan. He was also working on the same project right now at the University of Virginia, my longtime collaborator Iran Gafan. And for sequencing, Dimitri is helping us. And my 2 postdoc advisors, Alan and Paul, and funding from NIH and NSF. We'll be happy to take questions. Oh, sure. Here? Oh, so you have like 8 or 10,000 fly in a cage, right? You take a bowl of silica covered with a cheesecloth, put it inside, airtight that entire cage, and let's wait until 85% of the flies dies. So it, like every generation, it will change like every, when you are selecting, they are becoming more and more harder, so it will take time. So it like few hours when you start the selection experiment, it can go like half a day, then like full day, something like that. They have no access to food and water. Exactly, they have that food and everything, yeah. But desiccation is way faster than starvation. When you starve the fly, it takes like around 120 hours something. Exactly. These issues will there, but there is no other way. Like you can control that particular part inside the body or something. Yeah. Yeah. Yes. Thank you very much.